Welcome to Layla's Science Hub. Today, we're breaking down one of the most important topics in biology, nucleotides, DNA, and RNA. By the end of this video, you'll understand how these molecules are built, how they differ, and even how you can extract DNA using strawberries. A nucleotide is the building block of nucleic acids, like DNA and RNA. Each nucleotide has three parts: a pentose sugar, that's either deoxyribose in DNA or ribose in RNA, a nitrogenous base, adenine, thymine, cytosine, guanine, or uracil, and a phosphate group which links sugars together. If a nucleotide has more than one phosphate, it's called phosphorylated. For example, ATP is a phosphorylated nucleotide. Nucleotides link together with a phosphodiester bond, which is a strong covalent bond between the phosphate of one nucleotide and the sugar of the next. These bonds form through condensation reactions and are broken when polynucleotides are digested. Not all bases are the same size. Purines, such as adenine and guanine, are larger and have 2 carbon rings, whereas pyrimidines, such as cytosine, thymine, and uracil, are smaller and have 1 carbon ring. This matters because in DNA, a purine always pairs with a pyrimidine, so that the rings on the DNA ladder are equal in size for molecular stability. Here's a quick comparison. RNA, single-stranded ribose sugar bases A, U, C, G, and shorter, often just a working copy of instructions. DNA, double-stranded deoxyribose sugar bases A, T, C, G, and very long, carrying lots of genetic information. DNA is a double-stranded antiparallel molecule. Let's unpack that. The sugar-phosphate backbone runs in opposite directions. We call this the 5' end, where the phosphate is attached to carbon 5 of deoxyribose, and the 3' end, where it's attached to carbon 3. The 2 strands are joined by hydrogen bonds between complementary bases. Adenine always pairs with thymine, forming 2 hydrogen bonds. Cytosine always pairs with guanine, forming 3 hydrogen bonds. Mnemonic, G, cat. These hydrogen bonds make the DNA molecule stable, but also allow it to unzip during transcription and replication. You can see DNA with the naked eye by extracting it from strawberries. Here's how. Firstly, crush a strawberry in a bag. Then add washing up liquid, salt, and water to break open the cell membranes. Then add the solution to the strawberry mush and filter it. Finally, pour in ice-cold ethanol. The DNA will appear as a white, cloudy substance in the ethanol layer. Exam tip. Examiners love clear contrasts. For example, DNA contains deoxyribose, whereas RNA contains ribose. Or, purines are double-ringed, whereas pyrimidines are single-ringed.